Could ancient chewing gum give real insight into how our ancestors lived? Stay tuned as we dive into this real scientific study where researchers sequence DNA found in samples chewed by people nearly 10,000 years ago. Hey there, welcome to Scienceisyou. We're a growing channel with a mission to bring you fun reports of real biotechnology research papers and increase accessibility of scientific research. To support us, please subscribe and share with your friends. Thanks for watching. DNA, which carries the genetic code of bacteria, plants, animals, and people, is a stable material that can remain in samples for thousands of years. In this research paper, the scientists collected samples of pitch, a tarry resin from tree bark that our ancient ancestors appeared to have used as chewing gum. The authors of this scientific study obtained 3 samples of chewed pitch more than 9,500 years old from an archaeological site in the Scandinavian peninsula. The researchers discovered DNA from the ancient gum pieces, which they claim represents the oldest genomic material to date related to human activities in Scandinavia. But why ancient chewing gum? Why not just look directly at fossil remains? The authors were interested in getting at a couple of specific questions. First, what was the oral health of individuals during this time period, and what was their diet? Both of these can give clues to the lifestyle of these ancient civilizations. They explain that recent studies have shown that about 15% of all the microbes in your mouth, the oral microbiome, gets trapped and preserved in modern-day chewing gum. So they thought maybe the same can be true of ancient chewing gums, giving us a deep look into the microbiological world present in ancient times. As you probably know, definitely small pieces of food can get caught up in chewing gum as well, so it's logical to try to look at ancient diet through chewing gum also. So the authors sequenced the DNA found in the pitch pieces. This type of bulk sequencing of DNA from potentially many organisms to try to get an idea of what's in there is called metagenomics. Unknown DNA sequences can be mapped to sequences of DNA that we know correspond to certain organisms, sort of like matching fingerprints. This is often used to look at what types of microorganisms, bacteria, viruses, etc. are in a sample. So what did the researchers find in their ancient chewing gum samples? The authors found 51 individual bacterial genomes, most of which corresponded to bacteria that were pathogenic or disease-causing, or opportunistic, bacteria which can cause disease under certain conditions. Some of the bacteria the researchers identified were known to be involved in the dental disease periodontitis, a harmful gum infection. But just presence of pathogenic microbes doesn't necessarily mean you have a disease, and obviously they couldn't know if the people who chewed these gum samples 10,000 years ago actually had any symptoms. So they took it a step further and compared these ancient DNA samples to modern-day oral DNA samples. They were looking for signs of dysbiosis in their ancient samples. Dysbiosis means an imbalance in the microbial population in, say, your mouth. The human body isn't just human cells. We're full of microorganisms that enable healthy living, but if there is an imbalance, say too much of one type of bacteria that causes inflammation, it can cause health issues like perhaps periodontitis or dental disease. Comparing the proportions of certain microbes in modern-day oral samples to the ancient ones using computer-based models, they predicted over 70% probability that the ancient oral samples were indeed from individuals with periodontitis. The researchers here speculated that perhaps people of this time period would use their teeth more as, say, tools, increasing the odds of picking up disease causing oral bacteria. But in addition to looking at oral health of these ancient people, the researchers wanted to study their diet. In addition to microbial DNA, the scientists also found DNA from a number of plants and animals. A large amount was from hazelnut and wolf. But they also found DNA from red deer, brown trout, red fox, Arctic fox, eel, grass, and duck, and European turtle dove. The authors discussed that the DNA data they collected suggesting these plants and animals were part of the diet long ago are supported by archaeological finds like bird bones used for tools and remains of animals and hazelnut shells found at archaeological sites. Importantly, the researchers point out that although the findings of this DNA may give clues as to specific food sources in the Mesolithic diet, Their findings don't necessarily represent the whole variety of food sources consumed at the time, but may indicate what the people who chewed these pitch pieces had been eating shortly before. The researchers here believe that their results obtained from the gum DNA analysis paint a picture of a Mesolithic hunter-gatherer society in southwest Scandinavia that suffered from dental diseases. What do you think? Do you think any of these findings were surprising? Share in the comments. If you have enjoyed this video, please don't forget to subscribe to help out our mission of making real biotechnology research papers accessible and fun for everyone. And remember,